All right, everybody, welcome to another deep dive. Today, we're getting up close and personal with the Chiazzi test.
It's a classic for a reason.
Now, I know what you might be thinking. Bacteria, tests, agar slants. Sounds a bit like a snooze fest, right?
Don't worry, it's way more exciting than it sounds.
Exactly. This isn't just some dusty lab procedure, folks. This test is like a secret decoder ring for the bacterial world.
It's amazing what you can learn from just a little color change and a bit of gas.
Right. It's all about understanding the language of bacteria and why that matters. So let's dive in. What exactly is this Chiazzi test, and why is it such a big deal in microbiology?
Imagine you're a detective, right? But instead of a crime scene, you're dealing with, say, an outbreak.
Intrigue. Go on.
It could be food poisoning, a contaminated water supply.
Yeah.
You need to pinpoint the culprit, and fast. The Chiazzi test is one of your most valuable tools. It helps us identify specific types of bacteria, especially those pesky ones that like to hang out in our gut.
So it's like a bacterial lineup. We're narrowing down the suspects.
Precisely. And the beauty of it is you get so much information from just one test. It tells us all about a bacterium's metabolic capabilities, what they like to eat, what they produce as waste. It's all there.
You said eat and waste. That makes me think of the actual test itself. I know there's this special agar involved, right?
Right, you are. The Chiazzi agar slant, to be exact.
So what's in this magical agar that makes it so revealing?
It's all about the ingredients. Each one plays a very specific role. For example, we have glucose and lactose in there.
The sugars.
Exactly. They act like food sources for the bacteria. Some bacteria love both, some are pickier, and some, well, they want nothing to do with them.
So it's like we're setting a trap, seeing which bacteria take the bait.
That's a great way to put it.
This is already way cooler than I thought. Okay, so we've got the sugars. What else is on the menu?
Well, we also add peptone. That's like a backup food source in case the bacteria aren't into the sugars. And then we have some indicator chemicals like phenol red and ferric sulfate.
Okay, those sound pretty intense. What do those indicator chemicals do?
They're like our spies on the inside. They give us visual cues about what's happening in the agar. For instance, phenol red. That one changes color depending on the pH. So if the bacteria are producing acidic byproducts while munching on those sugars, the agar will turn yellow.
So that's how we know they're enjoying their meal.
Exactly. And as for ferric sulfate, well, that one helps us detect hydrogen sulfide.
Hydrogen sulfide, that rings a bell.
It's a byproduct that some bacteria produce. And if they're making it, we'll see this black precipitate forming in the agar. It's pretty cool to see.
Wow, it's like we're setting up this whole buffet for the bacteria and then spying on their dining habits.
That's a great way to think about it.
So we've got our bacteria happily settled in their agar buffet. Indicator chemicals are in place. What's next in this bacterial stakeout?
Now we play the waiting game. We incubate the agar slant, usually for about 24 hours, at just the right temperature that those bacteria love. It's like tucking them in for a nice cozy nap.
And then we get to analyze the results, right? Like, what are we actually looking for after the incubation? What kind of visual cues give us the intel we need?
Get ready for some pretty dramatic transformations. The agar itself becomes like a message board full of clues. And those color-changing pH indicators we talked about, they're the stars of the show.
So we're looking for color-coded messages from our microscopic friends. What do the different colors tell us?
Okay, picture this. You pull out the test tube, and the slant is red, but the butt, that deeper part, has turned yellow.
Okay, I'm picturing it. Red slant, yellow butt. What's it mean?
That, my friend, means these bacteria can only ferment glucose. They've used up all the easy-to-access glucose in that area, produced some acids, turned the agar yellow, but the lactose, not interested.
They're picky eaters. But hey, to each their own, right? So what about if we see a test tube where it's all yellow? Slant A and D, but totally yellow.
Now that's a sign of a real sugar fiend. Yellow slant and butt means those bacteria are happily fermenting both the glucose and the lactose. They like a complex meal, those ones.
They're not afraid of a little sugar rush. I like their style.
Yeah.
But what if? What if the agar is just stubbornly staying red? Like refusing to change color at all?
Then we might be dealing with some more discerning bacteria. Red slant and butt usually points to a lack of fermentation going on. These bacteria, they're strict aerobes. They need oxygen to survive, and they'd rather get their energy from that peptone we talked about earlier.
Fascinating. Okay, so the colors give us a good sense of what these bacteria are snacking on. But what about those cracks and bubbles you mentioned? What's the deal with those?
Those, my friend, are signs of a real gas-producing party. If you see cracks, bubbles, or even the agar lifting in the tube, That means those bacteria have produced some carbon dioxide and hydrogen gas during fermentation.
It's like they're, well, I don't want to say tooting, but
Hey, everyone's got to let off some steam, right? Especially after a hearty bacterial meal.
fair enough. Okay, so we've got our colors, our gas production. Anything else we should be on the lookout for in this microbial investigation?
Just one more thing. That black precipitate we talked about earlier. Now, don't be alarmed when you see it. It's not necessarily a bad thing. It just means our bacteria have produced hydrogen sulfide while breaking down certain compounds in the agar. Remember the baric sulfate? It's reacting with that hydrogen sulfide, creating that black iron sulfide we're seeing.
So it's like a little chemical reaction sideshow.
Exactly.
This is amazing. We can learn so much from just observing one little tube. But I have to ask, how do we connect all these visual cues back to specific types of bacteria? Like, can you give me some real-world examples?
Absolutely. Let's talk about E. coli for a second.
E. coli, everyone's favorite. Well, maybe not favorite, but
A very well-known one, let's say that.
right. So E. coli, common resident of our gut.
Mm-hmm.
Most strains are harmless, but then they're the ones that give you those nasty food poisoning episodes.
Exactly. Now, E. coli, they really love to ferment both glucose and D-lactose.
They're the ones going for the full buffet experience, huh?
That's them. So on the Chiazzi test, you'd expect to see that totally yellow tube, maybe even some gas production, depending on the specific strain.
So it's like their metabolic fingerprint. That's so cool. What about a bacteria that gives us a totally different result, though?
Let's see, how about Pseudomonas aeruginosa?
Pseudomonas. I feel like I've heard of that one.
It's a bit of an opportunist, this one, often found in things like soil and water.
Ah, okay.
And it causes infections, especially in hospital settings.
Oh, that kind.
Right. Now, Pseudomonas, it doesn't ferment glucose or R-lactose. So on the Chiazzi, you're likely to see a red or orange tube. It's getting its energy from that peptone we talked about, not from the sugars.
Wow. Okay, so we've got these bacterial calling cards, each one telling a story. This is way cooler than I ever imagined. But it makes you wonder, how is this knowledge actually apply to, you know?
It's not just about identifying the bacteria. It's about understanding what that means, what the implications are. Say a patient comes in with, I don't know, some kind of persistent digestive issue. The doctor could use the Chiazzi to help figure out what's going on.
Ah, so it helps them narrow down the potential culprits.
Exactly. And if it turns out the bacteria are producing that hydrogen sulfide, well, that could point to a more specific type of infection, maybe help the doctor target treatment more effectively.
So every bit of information from the test adds another layer to the picture.
Precisely. And this knowledge, it goes way beyond just diagnosing infections.
Yeah.
Think about food safety for a second.
Ooh, yes. Very important.
If we understand the metabolic profiles of these bacteria, like E. coli, Salmonella, you name it, we can develop better ways to prevent them from contaminating our food supply in the first place.
It's all about prevention.
Right.
That's incredible. We've gone from these simple color changes in a test tube to a deeper understanding of how to diagnose infections, keep our food safe. It's amazing how relevant microbiology is to our everyday lives.
It's everywhere, even if we don't always realize it.
Absolutely. And this is just the start, right? What about the future of all this?
Oh, the possibilities are endless. Researchers are always finding new ways to use these techniques. Imagine identifying harmful bacteria in real time, maybe even with some kind of portable device.
Oh.
Or using similar principles to study the microbiome, all those trillions of bacteria that live in our gut.
Right. They're super important for our health.
Exactly, and we're only just beginning to understand how it all works.
It's like opening a door to a whole new world. This deep dive has definitely blown my mind.
That's what we like to hear. It might seem like a simple thing, this Chiazzi test, but it really is a window into the fascinating world of bacteria.
Totally. So, to sum it up, from a little agar slant to a deeper understanding of bacterial metabolism, its impact on healthcare, food safety, I feel like we've learned so much.
It's been quite a journey. And like I said, we're just getting started.
So much to explore. Well, on that note, I think we'll leave everyone to ponder the amazing invisible world of bacteria. Thanks for joining us on this deep dive, everybody. Until next time.